All right, so thank you very much for the introduction. It's a real pleasure to speak here today. We've had so many stimulating talks on how the gut microbiome might influence human health and development, and it's my privilege to extend that conversation to the area of mental health and of cognitive development. It works. Wonderful. So I thought I'd start off by just giving you a sampling of papers, fairly recent, reporting altered gut microbiomes in various neurodevelopmental and psychiatric conditions. Autism is probably one of the most extensively studied areas, as well as major depressive disorder. Unlike the talk by Brett Finley this morning, at this point we still can't really say that any particular bacterial group is consistently implicated across various studies. There's a lot of heterogeneity. But there are differences. There have also been more recent reports of altered microbiomes in children with attention deficit hyperactivity disorder, individuals with schizophrenia, and individuals with anorexia. Now, obviously, demonstrating that there are differences is not the same as demonstrating causality. And we have to remember that people with these conditions interact with their environment in very distinct ways. So if we just take autism as an example, these children are often put on special diets by their parents. Although we don't have any medications that directly address the core symptoms of autism, these children are often on multiple medications to address associated symptoms like self-injurious behaviors or anxiety. And they just interact with their social world in very distinct ways that could actually cause a change in the microbiome. So our best evidence at this point that the gut microbiome actually may have a causative effect in the area of brain development is from animal studies, and particularly the study of germ-free rodents. So the most consistent phenotype that we see in the rodents is actually reduced anxiety in germ-free animals. Now, we tend to think of reduced anxiety as a good thing, particularly as I'm sitting here giving a talk. I'm thinking, oh, I wish I was less anxious. But if we're talking about mice, a small prey animal, I think you can actually think of this as a maladaptive behavior. If you're a mouse, you should be frightened of the world. Red here is reduced behavior, so they also have impaired recognition memory. They have reduced sociability. They have increased locomotion, increased self-grooming, which is often used as a sort of experimental model of repetitive behaviors. And these changes are accompanied by changes in circulating signaling molecules. So somewhat paradoxically, actually increases in cortisol reactivity, major stress hormone. And then in the brain, reductions in brain-derived neurotrophic factor, a number of neurotransmitters, altered neuronal transcription and neurotransmission, particularly in brain areas that are involved in these behaviors. So in neural circuits for anxiety, for social cognition. So things like the amygdala, hippocampus, and medial prefrontal cortex. The animal work has also shown us a number, let's see if this is actually going to work. It does. Of potential signaling mechanisms, so communication pathways between the gut microbiome and the brain, some of which have been discussed here already. But they can interact with enteroendocrine cells in the gut, changing the release of peripheral serotonin, which is where the majority of the body's serotonin is. They can interact, obviously, with the immune system, which we've heard about in exquisite detail in earlier talks. We've been particularly interested in the possibility that the microbes are actually producing neuroactive molecules that could ultimately transfer to the brain. So we've heard short-chain fatty acids mentioned earlier. These are capable of passing through the blood-brain barrier and could potentially alter gene expression in the brain during development. They also produce neurotransmitters like GABA. And finally, they can interact with, again, the intestinal cells and change signaling through the vagus nerve to the brain. So there are a number of potential pathways of interest. Now, to go into a little more detail in the area of anxiety, one of the things that's really interesting, here we have our germ-free animal. It's got, this is our normal animal, so there are its germs. It's got high anxiety levels. Our germ-free animal has reduced anxiety levels. If we recolonize with a normal microbiota for specific pathogen-free mice, but we do it late, around 10 weeks, this is post-weaning, we change many of the signaling pathways that I showed earlier, but you don't change the behavior. You still have the reduced anxiety phenotype. In contrast, If you recolonize earlier, around 3 weeks, which is around weaning, you alter the signaling mechanisms and you also normalize the behavior. So now they look like a normal animal. So as we've seen with many of the other phenotypes, there's evidence of a windowing effect, a critical period in early development. And perhaps that shouldn't be too surprising because as we've heard, infancy and early life is really the foundational period for establishment of the gut microbiota. We've obviously heard about, we've seen this figure before, we've heard about it in some detail. But as you can see here, the dramatic shifts in bacterial communities across the first 2 years of life, which are intimately related to changes in environmental exposures and feeding practices. So we see here that the kids start out with primarily being breastfed. Over time, we see the introduction of solid foods until we come to the point of cessation of breastfeeding. And this kind of mirrors that. So we have early communities that come from the vagina, that come from the skin, transient increases in actinobacteria, which includes the bifidobacterium, which are feeding on human milk oligosaccharides, as we heard in the last talk, and then increasing amounts of things like the Bacteroides here over time. Now, in parallel with these changes in the gut microbiome, this is the most dynamic phase of postnatal brain development. And I want to take a little time to kind of sell you on the importance of infancy for the brain and remind you that really the prenatal and the perinatal period is the foundation of our brain development. It's shown really nicely in this figure from Tebenkamp et al. What they've done here is they've taken gene expression patterns in the brain across the lifespan and grouped them based on their neurodevelopmental processes that they're engaged with. So we can see here very high levels of gene expression for cell proliferation, early in gestation, a mid-gestation peak in the genes involved in neurogenesis, and then increasing across pregnancy and across the first year of life, genes involved in synapse development, in dendritic development, and in myelination. And once they're up here, they're remaining fairly consistent across the rest of the lifespan. I also want to emphasize this is a very important period for psychiatric conditions, even psychiatric conditions that do not emerge till much later in life, like schizophrenia. What you see here is gene expression patterns. Again, the black line here represents the distribution across all of the genes in terms of whether they're highly expressed in the prenatal period or more highly expressed in the postnatal period. So here we have in red genes involved in ASD, and the pointer's not working quite right there, I don't know why, and genes involved in intellectual disability. The red line here is shifted to the right, so that's indicating an overabundance of genes with high prenatal and early life expression. In contrast, if we look at this middle bottom left, that's bipolar disorder, which pretty much mirrors the general distribution of genes in the genome. And finally, we have genes involved in neurodegenerative disorders in purple, so Alzheimer's, for example, and those genes are shifted to the left, so they're more likely to be expressed at high levels in the adult, which makes sense. I also wanted to draw in some anthropology here, given the theme of the symposium. So this is some work from Philippe Guns and Jean-Jacques Houlin, showing that the particular globular shape of the modern human brain really emerges in the first few years of life based on expansion, particularly of parietal lobes in the cerebellum, and this really distinguishes the modern human brain from the brain of other hominids, such as Neanderthals. I'm going to show you some data here that's from a very large and extraordinary cohort we have at UNC, the UNC Early Brain Development Cohort. This is about 1,000 kids who were recruited during pregnancy, received structural imaging scans of the brain around 2 weeks of age, came back at 1 year and 2 years for additional follow-up, and are now actually being followed up at 4, 6, 8, and 10. So this is the same kid who has been scanned as a neonate, a 1-year-old and a 2-year-old. And this picture is to scale. So you can immediately see the huge increase in brain size across the first year of life. So the brain is doubling in size. It's increasing by about 15% here. And in fact, some regions like the cerebellum are increasing by 200% in that first year. When you're born, you have about 35% of your adult brain volume. By the time you're 4 years old, you're at 96%. So it's all over by the time you're 4. Hopefully not, but the scale of change is very different later in childhood compared to these first few years. The other thing you might notice here is if we look at this T1 weighted image, if we look at the 1-year-old and the 2-year-old, the white matter, which is primarily consisting of the axons that are connecting different brain regions, shows up as white. But when we look at the neonate, it shows up as gray. And that's because the brain is primarily unmyelinated at birth. But by 2 years, you have an adult-like pattern of myelination. And that's important because myelinated axons carry information faster, but they are less plastic. All right, I also want to emphasize that this isn't just about gross growth. It's not just increases in size. There's tremendous spatial temporal complexity. So this here is a map of cortical surface area expansion across the first 2 years of life. In the first year, we see the red regions are the fastest growing regions. And these are really in primary sensory and motor cortex. So auditory cortex, visual cortex, sensory motor cortex. If you look at the second year, there's now a shift. And the fast-growing regions are really association cortices, particularly these parietal areas. We can see this if we look at other kind of neuroimaging modalities, which I did want to introduce because we may talk about some of these later. This down here is, I don't know if I have troubles with. So this is actually from a diffusion tensor imaging study. This is a way of looking at the maturity of white matter tracks using water diffusion, which is restricted in a myelinated axon or in a heavily structured axon bundle. So the red here is the neonate, and I'm not going to go into the details, but you can see the huge shifts in the diffusion parameters across that first year of life with much more modest changes between 1 and 2, and you see that for a variety of different metrics. This is a different way of looking at the brain. This is resting state connectivity. So these are infants, they're sleeping in the scanner. We can't really get them to do tasks, but we can look at how different brain regions are communicating with each other. If their blood oxygenation patterns are temporally correlated, that means they're having some kind of functional connection to each other. And what we've done here is these are spring plots. The colors represent particular canonical adult networks, for example, a visual network or an auditory network. And you can see that in the neonate, they're really spread apart. But as we get to the 1-year-old and the 2-year-olds, the colors are now coalescing. So you're actually seeing the organization of functional networks in the first 2 years of life. We can also look just at the overall connectivity within the different networks. And you can see that most networks show increases in connectivity within themselves over the first 2 years, with the exception of the sensory motor network, which actually decreases, which is what you see down here as well. So the question we really wanted to ask, and that I'll spend most of the talk today on, is does the microbiome affect neurodevelopment in humans during this critical period? And a lot of this work was done by one of my graduate students, Alexander Carlson, and also in collaboration with Andrea Azcarati-Pero, who runs our microbiome core at UNC. So the study design here is we had 89 infants 1 year of age drawn from this larger longitudinal study who donated fecal samples. We have structural MRI scans on 46 of these kids at 1 year of age, 27 at 2 years of age. People drop out over time a bit. We have 39 with functional MRI at 1 year of age. We didn't have quite enough at 2 years to look at that. And then we also have cognitive development on these children. So 86 of them had scores at 1, 69 at age 2. And the question we're asking is, are these things associated with each other? And this is work supported by the National Institutes of Health, as well as a local charity, the Foundation of Hope. So first of all, just for some technical details, we're using 16S rRNA sequencing here, focusing on the V1 and V2 variable regions, for those who are familiar and want to know about that. So we've seen a picture like this before. So each column here is representing a single child. And the colored regions here are representing different bacterial groups, in this case, different bacterial genera. So you can see just looking at this, the tremendous individual diversity and variability there is in this sample. So the first question we asked is, are there meaningful patterns in that data? Can we identify clusters of children whose microbial communities are more similar to each other than they are to other kids? And the answer was yes. We do see evidence for sort of 3 clusters of children in this particular sample. Now, obviously, these clusters are distinguished by complex communities, but for simplicity's sake, we're going to say one of these is characterized by relatively high levels of Faecalibacterium, one by relatively high levels of Bacteroides, and one by relatively high levels of an unnamed genus in the family Ruminococci. You can also get a little idea of the more complex communities in the bottom pictures, where you're seeing negative and positive correlations with that seed genera. So I think there are two different ways we can think about these. One, we were struck by the fact that applying similar techniques to studies of adults also shows you three clusters, one characterized by high levels of Bacteroides, one characterized by high levels of Rumentic-Coxsackie, and then one by high levels of Prevotella, which is something we don't see in the infant, which makes sense because that's a group really associated with kind of high fiber diets. So that's not what 1-year-olds are getting. So you could speculate that these may represent individual differences that might be consistent going forward in life. Another way to interpret them is to think that these are somehow indexing the maturation of the microbiome. So Bacteroides here is actually a more juvenile and more infantile typical group compared to the Faecalibacterium and the Rumentic-Coxsackie, which are really more adult-like bacterial groups. So this could also be about where kids are in their trajectory towards an adult-like microbiota. You may also be wondering how you end up in one of these groups. So we did take a look at some factors such as feeding practice and delivery method, and indeed kids who are breastfed and kids who are vaginally delivered are more likely to be in the central group here, the Bacteroides. But all of our subsequent analyses are controlling for those factors. So the headline here is that when we looked at cognitive performance, and we're using something called the Mullen Scales of Early Learning, which gives you a number of domain-specific scales. So it looks at gross motor skills, fine motor skills, visual reception, receptive language, and expressive language. And it also gives you an early learning composite, which is made by putting together fine motor, visual, receptive, and expressive without gross motor. And this is kind of like a baby IQ. So at 2 years of age, but not 1 year of age, we see a significant relationship between cluster and cognitive performance, with cluster 2 performing at a higher level than the other two clusters. And when you look at this at the domain-specific level, after correcting for multiple comparisons, you see that there are significant differences in receptive language, in expressive language, and although it's not significant, you see a very similar pattern for visual reception. So probably all three of those are contributing to the overall differences that we see. We also looked at what's known as alpha diversity. So this is kind of a measure of how diverse an individual's microbiome is. And we looked at a number of different metrics. Probably the easiest to understand is observed species, which is simply the number of species that were observed. We also use the Chow index, which is similar in the sense that it's looking at richness, how many different types of microbes you have. We also looked at something called the Shannon index, which is taking into account the evenness of the community. So in an oversimplified scenario, if you had 2 bacteria in your gut and you had 50% of one and 50% of the other, that would be a more diverse community than if you had 90% of one and 10% of the other. And we also look at phylogenetic diversity. So again, in an oversimplified scenario, if you had 2 microbes and they were closely related, that would be less diverse than if you had 2 that were distantly related. So if we look at the measures of richness and the phylogenetic diversity, we see the same pattern, where the kids with greater diversity actually have poor cognitive performance. And this was really surprising to us because, as has been mentioned earlier, increased diversity is often thought of as the good situation, where you have a community that's going to be more robust to perturbations. And indeed, there have been reports that alpha diversity in this age range was associated with positive health outcomes for things such as diabetes. We don't see that with the cognition. We have a couple ideas about that. One possibility, perhaps the more diverse community is actually harboring pathobionts that the less diversity community is not. Another possibility is that the more diverse community means less resources afforded to those subset of microbes that have beneficial effects on cognitive development. And perhaps the most speculative idea would be, again, since alpha diversity increases over time, that a longer period of development for the microbiome might actually be beneficial for brain development, maybe an extended period of plasticity. You may also be asking why age 2 and not age 1? So we have a couple thoughts about that. One possibility is simply that the age 1 scores are less reliable. A 1-year-old child has a more limited behavioral repertoire to look at in the lab. You're relying more on parental report. Another possibility, and we've seen this in some of our other work, is that sometimes the 1-year-old time point seems to pick up how fast kids are developing right then, but isn't very predictive of long-term differences, whereas the 2-year really is. It's actually correlated with 6-year IQ. And of course, the explanation we like best is the possibility that the microbiome at age 1 is actually changing those signaling pathways in a way that alters brain development between age 1 and age 2, resulting in the cognitive differences at age 2. And kind of backing up that, this is just a comparison of the change in Mullen scores from 1 to 2 years, where you see they're all very much grouped together, but that there's a greater increase in the high-performing cluster. Hello? There we go. So another question you might have is, what's actually different about these communities functionally? So we did take a look at that using a program called PyCrust, which basically estimates predicted gene functions in the different clusters. So here, cluster 1 is actually representing the baseline. Green is cluster 2, the high-performing cluster. Orange here is cluster 3, one of the lower-performing clusters. And this doesn't represent all the gene pathways that are predicted to be different, but some that I think are particularly interesting. So you can see here that the high-performing cluster has an overrepresentation of genes involved in the synthesis and metabolism of various vitamins and cofactors. For example, increased folate biosynthesis. We know that folate is supportive of brain development and cognitive development. Increased biotin metabolism, so this is a B vitamin, and increased lipoic acid metabolism. Lipoic acid is involved in a number of different cycles, including the citric cycle, which is a major source by which energy is extracted. So maybe this is a group that is creating more supportive environment, getting more energy out of food, providing more vitamins and nutrients. Perhaps even more striking, we see that C2 has less expression of genes involved in bacterial motility, in sporulation, in bacterial chemotaxis, and flagellar assembly. So that might be compatible with this idea that there are pathobionts in the less well-performing groups that are really activating and heading out to cause trouble. Another question you may have is, what are the neural circuits actually mediating that effect? So the first way we looked at it is we just looked at whether cluster membership or alpha diversity was related to brain volumes. And we looked at gray matter volumes in 90 different cortical and subcortical regions. And we did see a few changes that were significant after multiple comparison correction in some occipital lobe areas, in the bilateral caudate at year 2, alpha diversity related to amygdala, some precentral areas. I want to be very cautious about this because it's an exploratory analysis. With the exception of the caudate, they're not bilateral findings, they're distributed. So I think we need to be cautious about that until replicated. I want to get a little more excited about some recent findings we've had on functional connectivity in these children. So this is work we did with our collaborator Wei Gao at Cedars-Sinai and his postdoc Andrew Salz-Waddell. And to look at this, we took 2 different approaches. First, a lot of the rodent work has focused on the role of the amygdala, because this is a key node in circuits for anxiety and for social cognition. And indeed, in germ-free rodents, you see changes in the volume, in dendritic complexity, and in gene expression and monoamine expression in the amygdala. So the first thing we asked is, are there differences in connectivity from the amygdala to other brain regions that are associated with alpha diversity? Because it was a smaller sample, we didn't look at cluster membership. Some of the clusters were very small. So you can see here that there is, in fact, a large significant cluster which overlaps with the thalamus, which is inversely related to alpha diversity. And this is actually interesting because particularly the paraventricular thalamus is associated with the processing of fear reactivity. So although we did not look directly at anxiety-related behaviors, this suggests that the same thing that's observed in rodents may be observed in human children as well. And that's something we're going to be looking at in our current studies. We also took a more exploratory approach, where we essentially took a seed representing these various canonical adult networks and looked at how that related to alpha diversity. So you can see here that we do see a relationship in functional connectivity between the anterior insula. This is a seed for the salience network. The salience network kind of directs how you take in incoming stimuli and define them as important or not important. And it also actually recruits other functional networks based on what the incoming data is. So it's a very key network. We see an association with connectivity between that and anterior cingulate, which is also a really key node in emotional reactivity, fear, and anxiety. So again, this is leading back into the anxiety story. Now, neither of those connectivity relationships explain the relationship with cognition that we observed, which is not surprising. It's a completely different neural circuit. But we also found that alpha diversity correlates with functional connectivity between the supplementary motor area. This is a seed for the sensory motor network, which you can imagine for an infant is actually pretty crucial to their learning and exploration of the environment. And then the left inferior parietal area, which is an area that's involved in top-down attentional allocation, that's involved in social cognition, that's involved in language acquisition. So here we see a direct correlation with alpha diversity, and we also see that this connectivity predicts cognitive performance. It's associated with the Mullen cognitive composite. So this actually looks like a neural circuit that might be mediating the impact of the microbiome on cognitive outcomes. So we're pretty excited about that. Some outstanding questions. Are the observed associations between the gut microbiome and cognition causal? We can't address that. It's a possibility. But it could also be that the microbiome is essentially standing in as a proxy for some other factor that we haven't measured. Obviously, we're interested in what's going on with other phenotypes like anxiety. If the gut microbiome has a causal role in these changes, how do we actually go about promoting the optimal development of the gut microbiome? How do nutrients and feeding patterns impact colonization, which is something we're looking at in a new study? How do host genetics impact microbial colonization? So we've collected DNA on these children as well, because you can kind of visualize if the host genome is set up to accept certain kinds of bacteria, it might be quite difficult to shift that community in a long-term way. What's the critical period for intervention? How do we keep the microbiome from resetting to a less optimal community? And how do we even define an optimal community? Because as we've seen, the very things that I'm showing you were related to poor cognitive outcomes were associated with better outcomes for asthma. So how do you decide what an optimal community is when there may be no gold standard microbiome that's best for every phenotype you're interested in? And then finally, what signaling pathways actually mediate between the gut and the brain? There are a lot more questions, but those were just as many as I could fit on that slide. So this is a study we're actually carrying out right now. We're going to collect data on 140 children. These kids are coming in at 2 weeks of age. They're a minimally medically intervened population. They're all vaginally delivered. They're all breastfed until at least their first visit. None of them have spent time in the NICU. We're getting fecal samples. We're doing blood spots to look at particular signaling pathways. We take advantage of the blood spot as an adverse stimulus to actually look at their cortisol reactivity to stress. So we get saliva for that, and of course they're getting MRI scans of the brain. They come back at 1 year of age. We get all of these things again, but we also do some behavioral tasks focused on anxiety. So I scare children for a living, apparently. I didn't think I'd end up doing that. This is the strange situation paradigm. It's classically a test of attachment, but we're using it to look at fear reactivity in a novel situation. And it really mirrors the kind of activities that a kid does go through in a day when they're being introduced to a new relative or they're going to daycare for the first time. So they're coming into this new space, they get to sit down on the floor, play with a bunch of new toys, but then a stranger comes into the room. And at first the stranger just sits there, then he starts talking with mom, then he sits down on the floor and starts interacting with the child, and then mom leaves the child alone with the stranger for a period of time. So it's kind of an increasing level of strangeness, and we see how the kids react. And then we have the masks task, which is just like it sounds. The experimenter tries on a series of Halloween masks. This is one of the ones we use. We also have an alien, a witch, and a gorilla. and we see how the kids react to the mask. And it's interesting, because some kids think they're hilarious and will laugh and want to play with the mask, and other kids are very reactive. And we have some very specific rules, too. We never let a kid hard cry for more than 15 seconds without calling it. And if they have a really strong reaction to the first mask, we won't go on to a second or third mask. We have a particular, and I should say we also do cardiac recording during this, so we can look at their heart rate variability during a moderate stressor. We have a particular model we're interested in testing. This was based on the animal work. So our initial hypothesis, at least, was that low levels of lactobacilli, low levels of actinobacteria, increased levels of gamma proteobacteria, increased levels of Bacteroidetes would be associated with greater immune activation and with greater cortisol reactivity. And we were really intrigued by the fact that both of these pathways converge on this tryptophan metabolism pathway. They hit different enzymes, but they both have the same influence. They're kind of the synergistic effect, where tryptophan is diverted away from being converted to serotonin. So you get essential serotonin depletion. Serotonin is a major mood-regulating neurotransmitter. And you get an increase in these metabolites in the kynurenine pathway. So these are potentially neurotoxic metabolites. And then our prediction is that's actually going to change the growth of the amygdala, the hippocampus, and the medial prefrontal cortex, so core nodes in those anxiety circuits. It'll change amygdala to medial prefrontal cortex connectivity, and that's going to result in increased anxious behavior. So that's what we're going to be testing. And I want to finish up by coming back, one of the focus, right there at the end. So one of the focuses of this session was going to be kind of on interventional time periods. So I wanted to talk about clinical implications at the very end. And the big idea here is modulation of the gut microbiota a tractable strategy for supporting cognitive development and reducing risk for later psychiatric disorders? So we heard a little bit in the last talk about the situation where the microbe is the drug, as is the case in a probiotic. The microbe can also be a factory that produces a drug or an intervention. Obviously, many antibiotics are actually bacterial products. They're also used for cancer treatment. They're used for cholesterol-lowering drugs. So can we do the same thing with psychiatric outcomes? I'm not going to talk so much about it being a vehicle, but we also have the possibility that if we could identify pathobionts associated with poor cognitive outcomes, then you could also have it be a target killed by the drug. So I'll finish up there with my acknowledgments. My various colleagues at UNC, Cedars-Sinai, Hong Kong. I want to thank all the participating families, my own beautiful children feeding their microbiomes with chocolate and rice cereal. And I'll take any questions.